Biology 1108, presentation number 14, Animal Digestion and Nutrition. One of the first things we need to consider in talking about animal nutrition and digestion is the fact that animals come in a variety of types, shapes, and forms in terms of how they eat. We're going to be looking specifically at some point at phagotrophic animals, which include ourselves, and those animals that consume food items larger than themselves. Phagotrophic animals, or more officially known as bulk feeders, engage in different trophic behaviors. We'll look at that. They have a certain choice of diet, which we'll look at that. And they demonstrate digestive tract modifications. We will look at that. Trophic behaviors are the behaviors associated with eating. The word trophic means to eat, and so how the organism eats varies from organism to organism and group to group. We've recognized this in the laboratory. So we've got organisms that are referred to as suspension feeders, commonly referred as filter feeders, like the sponge at right, although you could also draw an arrow down to the whale at below, consuming large numbers of krill. Would also be an example of a filter feeder. Substrate feeders are organisms that eat the substrate in which they live. Deposit feeders are organisms that consume materials on the surface of which they live. Fluid feeders are organisms that consume their diet in form of some type of liquid. I see the hummingbird consuming nectar at right, although that could include mosquitoes. And then finally, I've got bulk feeders listed there, which are organisms that consume their food items in rather large portions. For example, humans, dogs, cats, things like that. Another aspect of trophic behavior is classifying the organism in terms of what it eats. Herbivores eat plants, carnivores eat animals, omnivores eat a bit of both, and detritivores consume living material that was once alive that is now dead, like, for example, the earthworm consuming organic material contained in soil that is ultimately derived from things that have died and decomposed and have fallen into the soil material itself. Another aspect of trophic behavior is a trophic relationship between what is eating and what is being eaten. The grazer, like the deer at right, is going to essentially remove bits and pieces of the food organism, the grass, without killing it. The food organism is not removed from the population. On the other hand, the predator, like the lion below right, is going to actually remove its food item from its prey population. And then finally, the parasite at left, which is loa loa, or the African eyeworm, is going to derive its nutrition from a host. As a rule, parasites do not remove their host from the population. It would not be in their best interests evolutionarily. On the other hand, the host could die as a consequence of secondary health issues arising from that case of parasitism. The food that that animal eats contains nutrients, which are molecules that will support the life of the animal, both at the level of the organism and at the level of individual cells. Now, nutrients come in a number of different categories, and on this slide and the following slide, we are going to note that there are organic versus inorganic nutrients, there are macronutrients versus micronutrients, and there are essential versus non-essential nutrients. This slide we're looking at right now will involve organic versus inorganic and macro versus micro. Here's the deal. Organic nutrients are molecules that are composed of covalently bonded carbon atoms. And those molecules, as a rule, are going to contain stored energy. So we can refer to them as calorie-containing molecules, or sometimes they're commonly called food molecules. So the animal who eats is going to be ingesting what we are going to generally call organic macronutrients, covalently bonded carbon molecules that are going to be required in large amounts. The word macronutrient implies required in large amounts. And so these would be the food molecules you and I are most familiar with: carbohydrates, fats and oils, sometimes called lipids, and proteins. On the other hand, we do require inorganic nutrients, and inorganic nutrients do not contain covalently bonded carbon atoms. And for the most part, inorganic nutrients would include water and salts, ions, also called electrolytes, things like sodium chloride and a host of other things like that. Now, the thing is inorganic nutrients could be required in large amounts, like water, like calcium, maybe sodium, but many of them are required in small amounts, which are the micronutrients, and those would be things that are far less common, like zinc or iodine, things like that. This next slide picks up from where we left off on the last one, and that is under the topic of organic nutrients. There are organic micronutrients, which we refer to as vitamins. Vitamins, as a rule, are not required in large amounts, even though they're composed of covalently bonded carbon atoms, and they are not a source of calories. Vitamins, on the other hand, perform some other very necessary jobs for us. They are building blocks for other compounds. They function as coenzymes, and they can function as antioxidant substances, protecting us from the dangers of highly reactive molecules called free radicals. Now, other classes of nutrients is where we bring up the difference between essential and non-essential nutrients. So nutrients can be organic, nutrients can be inorganic, they can be essential or non-essential. An essential nutrient is a nutrient that, if removed from the diet, will result in a decline in health quality, but if restored, will result in an improvement in health quality. Obviously, an essential nutrient is needed to survive. On the other hand, non-essential nutrients are not going to either improve or decrease health quality overall. Although you could actually find some benefit of consuming non-essential nutrients, and it might essentially improve your health in some respects, but it's not going to be a consideration in terms of whether you stay alive or not. So, for example, a non-essential nutrient for a human being might be a compound called lycopene, which is found in tomatoes. Lycopene is not going to make the difference between whether you live or die. If you don't like tomatoes and you don't eat tomatoes, you're not going to die as a consequence of not eating tomatoes and not getting this compound called lycopene. On the other hand, lycopene can function as an antioxidant, and as I had mentioned a moment ago, antioxidants are compounds that essentially combat highly reactive molecules called free radicals that can actually cause damage to your cells. So therefore, consuming lycopene, while it's not critical to your life, can improve your health, at least in preventing some damage that could ordinarily occur from these compounds called free radicals. Okay, digestive tract modifications. The digestive tract is that part of the animal, generally a collection of organs that are going to be involved in processing the food the organism eats in order to extract the nutrients it needs. The digestive tract begins with the mouth, where food comes in, and that might be associated with a variety of feeding appendages, mouth parts, teeth, and then the actual digestive tract itself, which is nothing more than a really, really long tube in most cases, and that tube might be regionalized. Parts of the tube might be modified for doing certain aspects of the digestive process. It might be of a certain length. Animals that consume plants generally have a longer digestive tract, whereas animals that consume meat have a shorter digestive tract. A lot of times it boils down to how difficult the food item is going to be to digest. And then finally, all along the length of the digestive tract will be a variety of glands that produces those digestive substances that will help break down that food and in some cases manage those nutrients from the digestive tract as they are absorbed into the cells of the digestive tract itself, and then finally transported to the cells of the body through whatever circulatory system that animal might have. As an animal eats, how is that food absorbed? How is that food digested? Well, in the sponges and the more simple animals, digestion is a process that occurs inside individual cells. That's what the term intracellular means. You're going to have an event of phagocytosis that brings the food item into individual cells, and the digestion process will actually take place in food vacuoles inside the individual cells. That would be a protist or a primitive type of mode of digestion. On the other hand, when you get a little bit more advanced, like the anemone at the bottom right of the slide, there's going to be a combination of intracellular and extracellular digestion. You will remember that anemones are a member of the phylum Cnidaria, which is the jellyfish, and they are characterized as having a gastrovascular cavity, or GVC. And that gastrovascular cavity can be a location where digestion will take place. So some digestion taking place inside of the cells and some digestion taking place in the gastrovascular cavity, which technically is outside of the individual cells. And then finally, in the most advanced organisms, like that cute little mouse right there, like human beings, like most other animals we are familiar with, the process of digestion is almost exclusively extracellular, taking place in a digestive tract that is devoted exclusively to the digestion of those food items and then ultimately the absorption of the individual nutrients, so they are then transported to the individual cells by the blood circulatory system. At this point, we are going to be focusing on the human digestive tract, and the human digestive tract is not much different from the digestive tract of any other vertebrate we might encounter, be it a dog, be it a cat, be it a cow. Humans, animals, as a rule, have a complete digestive tract. That means you've got a mouth at one end and an anus at another. As a rule, most animals are discontinuous feeders. In other words, they do not eat constantly all day long. They eat at specific occasions, and their digestive tract is regionalized. So specific regions of the digestive tract are modified for doing certain things. For example, we got a mouth that processes food as it comes in. We have a small intestine that does a tremendous amount of absorption, and we have a large intestine that basically absorbs water. And at the very end of our digestive tract, we have a rectum that collects that waste before it's expelled from the body. All along that digestive tract, humans and animals have a variety of accessory glands that produce substances that aid in the process of digestion. So in taking our little trip through the human digestive system, we begin with the mouth. The mouth is a regionalized part of digestive tract, and it takes care of some specific things. It's going to take care of two basic aspects of digestion. Digestion can be mechanical, digestion can be chemical. Mechanical digestion is the physical pulverization of food material, and what that does is it increases the surface area of that food material so you can actually be more effective in terms of breaking down that food material into the individual absorbable nutrients that we need in the first place. So our teeth and our tongue are going to be involved in the mechanical digestion process. We're going to take that food, we're going to break it down physically into smaller and smaller bits, increasing surface area so we can better accomplish chemical digestion. Now, chemical digestion is using enzymes to break down that food material into the individual absorbable nutrients we need. So you eat a hamburger, You chew it up. The hamburger is ultimately broken down in little bits so that the various enzymes in your digestive tract can basically break that food material down into the sugars, into the amino acids, into the fat molecules that are ultimately going to be absorbed and transported to your individual cells. So in the mouth, chemical digestion is pretty much limited to the breakdown of starch. You will see immediately below that bullet point, the bullet that says salivary amylase. We produce this stuff called saliva. Saliva contains an enzyme called amylase. Amylase is an enzyme that breaks down starch. You'll remember that starch is a really, really long polysaccharide composed of individual glucose molecules linked together. If I can break apart that starch, if I can break the glycosidic bonds that link those glucose molecules together, I can absorb those glucose molecules into my bloodstream and transport them and deliver them to my individual cells to support the process of cellular respiration. It's how we stay alive. So chemical digestion in the mouth is limited to the digestion of carbohydrates or starch. Notice we don't do any fat digestion and we don't do any protein digestion. Once we get done with the chewing process and we've got this food reduced to this soft, moist mass, that soft, moist mass is going to be pushed to the rear of the mouth. We're going to form this thing called the bullous, which is the soft, moist mass of food that will ultimately be swallowed. Technical term for the little, soft, moist mass of food that is going to be moved from the mouth into the esophagus through the act of swallowing is called the bullous. One of the problems we have at the posterior of our mouth, which is called the pharynx, is we have two openings found there. One opening to the esophagus that leads to the stomach, and a second opening, which is the larynx or the voice box that ultimately leads to the windpipe, commonly called the trachea. Somehow we need to make sure the food gets from the pharynx into the esophagus and not into the windpipe. If you take your fingers and you put them against your throat and you swallow, you will feel your Adam's apple rise and fall. Now, the Adam's apple is part of your larynx. It's basically nothing more than a cartilaginous box that sits at the top of your windpipe. So during an act of swallowing, the action of the larynx is that larynx is going to rise and fall. Now, as it rises and falls, it is going to come in contact with a little lid-like structure called the epiglottis. At the top of the larynx is a little opening that is officially called the glottis. It is the opening from the pharynx into the larynx. It is the opening through which air moves in and out when you breathe. But the fact of the matter is during an act of swallowing, you want to close that thing up. So as you swallow, the larynx rises and it contacts this structure called the epiglottis. Epi, the prefix, means on top of. It's like a little lid. It's like one of those little trash cans when you push the little pedal and the lid opens and closes. In this case, the epiglottis is the lid that is closed on top of the glottis so that during the action of swallowing, the bullous, instead of moving into the larynx, instead of moving into the trachea, will then move into the esophagus. And you will see that in a series of illustrations at right. Once that bullous gets to the esophagus, there is a ring-like muscle at the top of the esophagus called the esophageal sphincter. A sphincter is any ring-like muscle, and it actually regulates an opening. So when the sphincter relaxes, the opening is in the open position, and if the sphincter contracts, the opening is in the closed position. So the esophageal sphincter that's located at the top of the esophagus is going to basically relax, at which point the bullous will easily move from the pharynx into the esophagus. The esophagus is a region of the digestive tract that has absolutely no digestive function. It is nothing more than a tube that connects the pharynx with the stomach. We had mentioned at the top of the esophagus there's this thing called the upper esophageal sphincter. The tube itself, once the esophageal sphincter opens, is going to aid in the propulsion of the bolus downwards. You'll see the illustration at right. The esophagus is a muscular tube. It is composed of a combination of striated muscle tissue and smooth muscle tissue. It is under involuntary control, and you will notice there is this wave-like process of contraction. It's officially called peristalsis that will actually push the bolus downwards through the esophagus. Now, in human beings, the esophagus is vertically oriented, so gravity is going to play a role in getting that food downwards, but the majority of the mechanism to getting that bolus down to the stomach is through a process called peristalsis. Now, one other comment about this thing called peristalsis, and that is, peristalsis is a characteristic feature not only of the esophagus, but I'm going to find it in the stomach, I'm going to find it in the small intestine, and I'm going to find it in the large intestine. It is the major mechanism of how we actually push food through all of those regions of the digestive tract. Finally, at the base of the esophagus, there is another sphincter muscle called the lower esophageal sphincter that, when relaxed, will allow that bolus to drop into the stomach cavity, and then it will close again. Let's take a look at the stomach. The stomach is a multi-layered structure. The outermost layer of the stomach is called the serosa, which is a layer of connective tissue that functions as a structure against which the muscles can contract. Immediately below the serosa is multiple layers of muscle called the muscularis, and inside of that is the lining of the stomach, which is called the mucosa. And the mucosa is where we have the glands that produce digestive substances. We'll also produce mucus that will aid in the lubrication of that food material so it passes through the digestive tract easily, but at the same time, the mucus produced will also protect the stomach lining as well. Those glands are located in deep pits within the mucosa that are called gastric pits. The mucosa is pretty thick, and those gastric pits are lined with mucus cells that produce this goopy stuff called mucus. It will contain specialized cells called chief cells that will produce an inactive enzyme called pepsinogen, more of that in a moment, and then parietal cells that produce a strong acid called hydrochloric acid. The stomach also engages in both of those types of digestion that we mentioned in context with the mouth. The stomach is mechanically digesting food. As it contracts, it breaks that food down into smaller and smaller bits, and the stomach is going to engage in some form of chemical digestion. Now, as far as the chemical digestion is concerned, the stomach will actually digest food materials simply through the action of that strong acid, hydrochloric acid. And then again, we're also going to engage in some protein digestion in the stomach. Now, you'll remember that chemical digestion in the mouth was limited to starch. In stomach, we're going to be a little bit more broad, but as far as the stomach's concerned, one of the most important things it does is it begins the breakdown of proteins. You break down proteins into individual chains of amino acids that are called polypeptides. It is not a complete form of protein digestion because we ultimately want to break down proteins into individual amino acids, which can then be absorbed, which can then be transported to the blood circulatory system. But in the stomach, we're going to start the process. We'll take a big, giant protein and we'll break it down into individual polypeptides. And that is accomplished by an enzyme called pepsin. Now, you remember just a minute ago, I said that the stomach mucosa produces an inactive enzyme called pepsinogen. Pepsinogen needs to be activated in order to digest proteins. Now, this makes sense because since the stomach is primarily protein in terms of its structure, it's a muscle, the last thing you want to do is digest the protein in the stomach wall. So we're going to go ahead and make an inactive enzyme called pepsin, and we are going to wait to activate that pepsin until it gets into the cavity of the stomach. And at that point, we can break down the proteins in what we've eaten instead of breaking down the proteins in our stomach wall. Now, if you look at the second major bullet on the slide, it says that in our stomach, we're going to have the secretions, the mucus, the hydrochloric acid, the pepsinogen, which is called gastric juice. We're going to have the food, which is in the form of the bolus, and the mixture of that creates kind of a soupy stuff called chyme. Chyme is the contents of the stomach. It is a combination of mucus, hydrochloric acid, pepsinogen, and the bolus that has recently been swallowed. That mixture is ultimately going to be moved from the stomach bit by bit by bit into the small intestine, and the region where the stomach connects to the small intestine, as illustrated in the picture at bottom right, is called the pylorus. The pylorus is the place where the stomach attaches to the small intestine, and the pylorus is characterized by a ring-like muscle called the pyloric sphincter that will actually open and close, regulating the input of chyme into the small intestine bit by bit by bit. So it's going to be a regulated introduction of this partially digested material into the small intestine so we can complete the digestion process and be really, really effective in terms of absorbing these nutrients. All right, just a moment ago, I mentioned this stuff called pepsin. And pepsin is the active form of pepsinogen, and pepsin breaks down protein. Well, we are presented with a problem because since our stomach is made of so much muscle, the last thing we want is to digest the musculature of the stomach. But it sometimes does happen. Most of us heard of something called ulcer, which is officially known as peptic ulcer, which is a localized erosion of the stomach wall. And you'll see it at right. You'll see the guy in pain. You'll see the picture of what looks like a little itty-bitty inflamed volcano in the lining of the stomach. If you have a problem and the pepsin starts to eroding the stomach, it leads to this problem. Safeguards against peptic ulcer are built into our bodies. We produce all that mucus we mentioned before. We produce the pepsin as an inactive enzyme called pepsinogen, and for the most part, that works. Turns out there is also a certain bacterium that is always associated with peptic ulcer. It's a bacterium called Helicobacter pylori. Once the chyme moves from the stomach into the pylorus, it will be regulated in terms of its introduction into the next stage of the digestive tract, which is the small intestine. The longest section of the digestive tract, the small intestine itself, is divided into three regions: the duodenum, the jejunum, and the ileum. The small intestine is a hotbed of activity. It's going to be a place where digestion will continue. It will be a place where we will ease into and dominate the process of absorption. As we move through the small intestine, we move the chyme from the stomach into the duodenum, where digestion will occur in significant amounts. That's why I got the capital D there. Absorption in smaller amounts, but as we shift from the duodenum into jejunum, you will see that we shift in what we're actually doing. Digestion becomes less of an issue, but absorption becomes a major issue, and the same is true with the ileum. We're decreasing the amount of digestion we're taking place because we've done most of it at this point. And we're improving the amount of absorption, which is really where the rubber hits the road. This is where we really gather all these nutrients we need, and we move them into our blood circulatory system, and we get them transported to our individual cells. Here's a close-up view of the relationship between the stomach and the duodenum. And what we need to know is that duodenum receives input from two different places. Duodenum is going to receive input from the gallbladder. You'll see the gallbladder is a little green sac that lies underneath the liver, and it connects to the duodenum through a little tube called the bile duct. Bile is a digestive material. We'll talk about it in a minute. It's produced by the liver. It is stored in the gallbladder, and that gallbladder is going to release that bile as we need it into the duodenum. In a nutshell, bile is going to help us to digest fats. But we also see that the duodenum is going to receive input from the pancreas, which is a gland that lies immediately below the stomach, and its input is going to be carried through a tube called the pancreatic duct. There's actually several of them that leave the pancreas that ultimately connect into a primary tube that enters the duodenum. Make sure you know that the duodenum receives input from the gallbladder through the bile duct and from the pancreas through the pancreatic duct. Let's focus on the gallbladder. And let's focus on this stuff called bile. Bile is a material that's produced by the liver. It is stored in the gallbladder, and its job is to emulsify fats. One thing you might remember from once upon a time is that fat is a non-polar material. It does not mix with water. And so what we need to do in our digestive system, which is essentially an aqueous or water-based system, is we need to create a situation where the fat that we have eaten is broken down in little bits, and we're going to maintain those fat globs as separate, distinct little bits, maintaining an increased amount of surface area so that we can actually effectively reabsorb that fat. Bile does that. Otherwise, the fat globs would actually coalesce into one big giant fat glob, and we would not be able to effectively digest the fat. Now, the thing is sometimes there are problems associated with bile, and that is bile can crystallize in the gallbladder, producing things called gallstones. One of the major components of bile is cholesterol, and so gallstones, which would be a problem that a person would need to have their gallbladder removed, is nothing more than the accumulation of crystallized cholesterol in the gallbladder. Okay, let's take a look at the pancreas. The pancreas, you remember, produces materials that are going to be conducted to the duodenum through the pancreatic duct. The pancreas is kind of a unique thing. It is a gland that is both an exocrine gland and an endocrine gland. An exocrine gland is a ducted gland. It is a gland where the substances produced by the gland will exit through a discrete duct. On the other hand, we got plenty of glands in our body that are called endocrine glands. Now, an endocrine gland is a ductless gland. So some of the substances produced by the pancreas, namely insulin and its compatriot hormone called glucagon, are going to be produced by the pancreas as an endocrine gland. The substances actually diffuse from the gland surface. So you want to remember that the pancreas is a gland with a split personality. It's part exocrine gland and it's part endocrine gland. Now, digestively, the pancreas functions as an endocrine gland. All of the digestive substances listed on this slide will exit the pancreas through the pancreatic duct. Now, in each of these cases, I'm going to need you to know the names of these digestive substances and what they do. So we'll see two of them up here called trypsin and chymotrypsin. Those two substances are very, very closely related in terms of what they do. And they are enzymes that are responsible for breaking down proteins into polypeptides. In other words, trypsin and chymotrypsin do the exact same thing that pepsin was doing in the stomach. You might note, therefore, that trypsin and chymotrypsin are going to be engaged in a partial protein digestion. We'll talk a little bit about how we actually complete the process of protein digestion in a little bit. The pancreas is also going to produce an enzyme called pancreatic amylase, which will continue the process of breaking down starch. It will produce a substance called pancreatic lipase, which is an enzyme whose job it is to dismantle fats. And then the pancreas will produce a hodgepodge of enzymes called nucleases, which will be breaking down DNA and RNA that are present in the foods we eat. Finally, the very last thing the pancreas does produce is a material called bicarbonate, which is not a digestive enzyme. Bicarbonate's job will be to readjust pH as we move from the stomach into the small intestine. Now remember this, the chyme is high in hydrochloric acid, which means chyme is going to have a very, very low pH. The contents of the stomach is a pH somewhere around 1 or maybe 2. Once we move in the pancreas, we need to bring that pH up to about 4. So bicarbonate does that. It neutralizes the acid in the chyme so that we bring the pH up to a point where trypsin, chymotrypsin, pancreatic amylase, pancreatic lipase, and those nucleases can effectively do their job. You'll remember that in the small intestine, the duodenum is followed by the jejunum, which is a relatively long section of the small intestine. The jejunum is really where the far majority of absorption will take place. So we've done the digestive process, and we're moving to a point in the digestive stream where we've got a bunch of nutrient molecules that are able to be absorbed. Well, the thing with absorption is going to be directly related to the amount of surface area we've got in the small intestine. So we're going to take this time to talk about surface area modifications. The small intestine is like a big long hose, but unlike a hose, the interior surface of the small intestine is highly modified to increase surface area. If you looked at the inside of a hose, there's a minimum amount of surface area inside the hose. If you take a look at the jejunum, there's a tremendous amount of surface area modifications going on. Let's take a look. If you take a look at the illustration on the right that is labeled Anatomy of Small Intestine, you will see that the interior of the small intestine includes folds, and those folds are coated with eensy-weensy, teeny tiny little finger-like projections called villi. And every single individual cell of the villus, that is, the cells that are covering the villi, are coated with their own little teeny tiny microscopic finger-like projections called microvilli. So we've got 3 different levels of scale as far as surface area modification is concerned: folds, villi, and microvilli. Let's focus on the villi. Each villus is a vascularized structure, and you will see that each villus contains blood capillaries, but will also contain a branch of the lymphatic system. And the difference is that the blood vessels, that is the capillaries, are going to receive input of water-soluble materials like sugars and amino acids. On the other hand, fats, which are nonpolar, are going to move directly into the lymph vessel. Now, each villus contains a branch of the lymphatic system, and that branch of the lymphatic system is called lacteal. So lacteal is a teeny tiny little lymph vessel that's found in each villus. Each villus is basically where we absorb most of our nutrients, and they will be moving from the digestive system into ultimately the blood circulatory system. I will mention this. At some point in the body, the lymphatic system does interconnect with the blood circulatory system, so those fats will ultimately get into our bloodstream, but the basic idea is fats are absorbed into the lymphatic system first. Now, one other mention here, and that is a bullet point that is referring to something called carboxypeptidases. Carboxypeptidases are a host of enzymes that basically complete the process of protein digestion. You'll remember that in the process of protein digestion, we're breaking down proteins into polypeptides. That's what pepsin did. That's what trypsin and chymotrypsin did. But we mentioned that was an incomplete protein digestion. Somehow we got to break down those polypeptides into individual absorbable amino acids, and that's what carboxypeptidases do. Carboxypeptidases are found all throughout the jejunum. They're made by individual cells, not by individual glands, and their job is to break down those polypeptides into individual absorbable amino acids. From the jejunum, we move into the ileum and pretty much continue on the same thing we've been doing. But the ileum is unique because it has an increased number of specialized mucus-producing cells called goblet cells. And this mucus will be produced in anticipation of the large intestine. What's going to happen is the large intestine is going to be in place where we will dehydrate this material. The material is basically waste at this point, and we need to be able to lubricate it so it can move through the large intestine with minimum amount of problem. So the ileum is basically going to be largely concerned with lubricating this material so it will move easily through the large intestine. But once again, we'll just understand that the ileum will also be in the process of absorbing those nutrients that we've liberated through the process of chemical digestion as we've been moving through the duodenum and as we have been moving through the jejunum. From the small intestine, we move into the large intestine. This particular slide will look at the parts of the large intestine. You could certainly Google this to get a better idea of the anatomy. This particular illustration might not be the greatest in the world. Where the large intestine meets the small intestine, we have a blind end sac called the cecum. It's where our appendix is located. And then the large intestine itself, which is commonly referred to the colon, has 4 different sections to it. The ascending colon, which rises, the transverse colon, which moves from right to left across the top of the abdominal cavity, the descending colon, which moves downwards, and the sigmoid colon, which is like a little S-shaped turn at the very end of the large intestine where it connects to the rectum. And the rectum is where fecal material is stored prior to elimination through the anus to the outside of the body. So what does the large intestine do? It's primarily going to function in the dehydration of waste material. Every single day, you and I contribute almost 8 liters of liquid to our digestive stream. That's 8 liters of water. That's 8 liters of stuff that's basically found in our saliva and all of the various mucus and enzyme secretions from beginning all the way to the end. If we lost that 8 liters on a daily basis, that would be a catastrophic loss of water. So what we're going to do is we're going to get that water back. So that's what the bullet point dehydration of waste material is going to involve. We are also going to absorb a bunch of things that are dissolved in that water. There's still a hodgepodge of salts we could get. There's still a hodgepodge of water-soluble vitamins we might be able to get. Water-soluble vitamins would include vitamin C and the B vitamins. And then finally, once we get all of that dehydration done, and once we get all of those hodgepodge of water-soluble things absorbed, what we got left is waste material that we can basically store prior to elimination. Our large intestine is a really great place for bacteria to live. And so every single one of us maintains a pretty darn rich community of colonic bacteria in our large intestine. Sometimes those bacteria are referred to as enteric bacteria. That's E-N-T-E-R-I-C. But there's enough of them that on a daily basis, 1/3 of the dry weight of the fecal material we pass is composed of these bacteria. They live on us as neighbors in a lot of ways. They're endosymbionts. They break down things that are normally undigestible for us. They are responsible for formation of certain vitamins, a certain amount, for example, of the vitamin K that you use is made by colonic bacteria in your large intestine. There's another vitamin called biotin, and I guess I should mention that one of the undesirable aspects of colonic bacteria is a lot of times when they do their thing, they produce a gas called methane, which contributes to that unfortunate human thing called flatulence. Now, one other point is that colonic bacteria sometimes get from our intestines out into the environment, and if they are found in water supplies, they are commonly referred to as coliforms. If you've ever taken a look at a water test report, either from a well or from a city or community system, they will mention coliforms at some point in that report, and it's just essentially some report of the number of these bacteria, and potentially they're disease-producing bacteria that might be found in a water supply. I had mentioned at the very beginning of this presentation that some animals demonstrate shorter digestive systems and some animals demonstrate longer digestive systems, and then sometimes there's some variation in the components as well. Let's take a look at some general differences between herbivores versus carnivores. Herbivores and carnivores have differences in terms of teeth. Carnivore teeth are generally modified for tearing and cutting, whereas herbivore teeth are modified for crushing. There's going to be a difference in terms of digestive tract length. Herbivores, as a rule, have a longer digestive tract, owing to the fact that plant material is a lot more difficult to digest. Carnivores have a shorter digestive tract length. There will be some difference in the regionalization of the digestive tract. In particular, in herbivores, the cecum will be larger and the colon might be larger to basically facilitate additional digestion that might be carried out by bacteria. And then finally, there's the presence of certain microorganisms that might be found in the digestive tract as well. I had mentioned bacteria in many organisms, but I should also mention that in ruminants, which would be animals like cows and deer and horses and things like that, there are some single-celled protozoans found in the digestive system that do much the same thing for them as the bacteria do for us. That's what that last bullet point refers to, symbiotic and commensalistic associations of organisms found in the digestive tract. Okay, so this slide is nothing more than a visualization of the content that I just mentioned in the context of the last slide. Herbivore on the left, carnivore on the right. You see some basic differences between the digestive tracts of the two. On the next couple of slides, we'll take a look at what happens to these nutrients after we absorb them. Let's take a look at carbohydrates first. These are sugars. They are water soluble, so therefore they are generally absorbed into the blood. And there's this hormone called insulin that gives our cells permission to absorb sugar from our bloodstream, but also gives our liver permission to take that sugar from our bloodstream and store it as glycogen. So in some senses, we're going to need to regulate our blood sugar. As we eat, blood sugar levels go up. In between meals, blood sugar levels go down. Let's see what happens here. In terms of when we eat, blood sugar levels go up. So what we have to do is we have to encourage cellular uptake by our cells, and we're also going to want to store that extra blood sugar as glycogen. That's what that word glycogenesis means. Glycogenesis means creation of glycogen. It's how we store glucose. In between meals, our blood sugar levels are going to go down, so what we might want to do is we might want our liver to do the exact opposite. Instead of storing blood sugar, why don't we go ahead and break down the glycogen and release it? And that is encouraged by a hormone called glucagon, and the process of breaking down glycogen is called glycogenolysis. And then finally, during periods of fasting, when you're either intentionally dieting or maybe when you're asleep during long periods of time, at some point you might run out of glycogen, at which point your liver might want to be able to make some glucose from scratch, and that's what's referred to as gluconeogenesis. What we can do is we can use amino acids as a raw material, and we deaminate the amino acids, remove the amino group, and what's left over we can use as a building block for making new glucose. Okay, so I know on the previous slide there was a lot of information, and it would have been nice to be able to have this covered in a face-to-face class because I got to draw a bunch of stuff on the board. But what I'm going to do is I'm going to use this particular slide as an opportunity to indicate to you what is going to be asked on an exam. So here's what you need to know. In terms of managing sugars, our liver is going to be involved in doing three different things. Make sure you write down each of these points and be very careful to know them in anticipation of an exam. The first thing that the liver does is something called glycogenesis. Now, glycogenesis is the creation of glycogen. Genesis means to create, glyco means glycogen. So after we've eaten a meal, our blood sugar levels are high, and what we're going to want to do is we want to store those sugar molecules as glycogen, and the liver is going to be involved with that. So glycogenesis is going to result in a decrease in blood sugar levels. The other thing I need you to know about glycogenesis is that it is encouraged by a hormone called insulin. So you eat a meal, you produce insulin, and your liver stores the excess blood sugar as glycogen, and your blood sugar levels go down. Now, the second thing that the liver does is called glycogenolysis. Now, this happens between meals. Your blood sugar levels are low, but your liver's got plenty of glycogen. So what it does, it breaks down that glycogen. Glycogenolysis, the word lysis means to break. So we're breaking glycogen. Your liver releases sugar into the bloodstream. So glycogenolysis means that blood sugar levels go back up. And glycogenolysis is also going to be encouraged by a hormone. Now, this hormone is called glucagon. Insulin's produced by the pancreas, and glucagon is produced by the pancreas. So once again, in between meals, you need some blood sugar. Your liver unloads it by breaking down glycogen, and that is stimulated by this hormone called glucagon. Finally, the liver sometimes runs out of stored glycogen. This might be during times of dieting. This might be during times of laying in the bed when you're asleep. But the bottom line is, even though your liver runs out of glycogen, your cells still need that sugar. So what your liver can do is it can actually build new glucose from raw materials, which would be amino acids. That's why we call this gluconeogenesis. Genesis means to create, gluco means glucose, and the word neo means new. So it's creation of new glucose. We're going to use amino acids as a raw material. If I deaminate an amino acid, what I'm left with is a 2-carbon compound. And if I take 3 amino acids, that means I deaminate 3 amino acids. I get 3 2-carbon compounds. That gets me 6 carbons, which equals 1 glucose molecules. It's just a way that the liver gets creative in terms of trying to be able to provide you the sugar you need. Turns out that gluconeogenesis will also result in an increase in blood sugar levels. And it turns out that gluconeogenesis is encouraged by glucagon, that same hormone that encouraged glycogenolysis, and it is produced by the pancreas. So at the bottom of the slide, you'll see the pancreas is responsible for making these two hormones, insulin and glucagon. Insulin and glucagon can be described as antagonistic hormones because they do the exact opposite. Insulin decreases blood sugar levels because it encourages cellular uptake of glucose, because it encourages glycogenesis in the liver, and glucagon increases blood sugar levels because it stimulates glycogenolysis by the liver or gluconeogenesis by the liver. Regulating blood sugar levels is an important daily activity for our bodies. But sometimes that doesn't quite work out. every single one of you has heard of a disorder called diabetes and are probably aware of the fact that individuals with diabetes do have elevated blood sugar levels. Elevated blood sugar levels has everything to do with insulin. So we will note on this slide there are actually 3 different types of diabetes I need you to be aware of. One is called type 1 or juvenile onset diabetes. One is called type 2 or adult onset diabetes, and the third is called gestational diabetes. The basic difference between the 3 types has to do with the nature of the insulin issue. In type 1 diabetes, juveniles is generally a result of an autoimmune disorder, whereas the body actually destroys cells in the pancreas that produce insulin. Therefore, the juvenile does not produce enough insulin to sufficiently lower blood sugar levels on a daily basis. In this case, the individual generally has to have an insulin pump or take regular insulin injections in order to moderate the disease. In type 2 diabetes, you've got an adult onset situation where the individual has probably chronically ingested large amounts of sugar over the majority of their lifetime. As that happens, the receptors on that individual's cells become less and less and less sensitive to insulin. So the more sugar you got, the less the insulin has an effect on that, which means the individual has progressively higher and higher and higher sugar levels in their blood, even though they're producing higher and higher and higher amounts of insulin. In other words, they're getting less bang for their buck as far as the insulin is being produced. These individuals generally need to diet, they need to moderate their intake, and they may, in fact, have to take insulin as well. Gestational diabetes is a rather rare situation that arises in pregnant females and generally shows up early in the pregnancy, but once the baby is born, the situation resolves. It's just important to note that pregnant women who do have gestational diabetes are often at an increased risk of developing type 2 diabetes later on in life. Let's take a look at what we do with lipids after we absorb them in the digestive system. You'll remember that lipids would include triglycerides, sterols, and some other nonpolar substances. And the problem with those nonpolar substances, in many times, they are not very easily transported in our aqueous-based bloodstream. So in terms of triglycerides specifically, absorption is going to involve the glycerol part of the triglyceride being absorbed directly into the blood vessels, since glycerol is a water-soluble substance. But the fatty acids, on the other hand, are going to be absorbed into the lacteals, or the lymph vessels, found in those villi that we were talking about not too long ago. Once the fatty acids get into the lacteals of the lymph vessels, they are assembled into itty-bitty little delivery trucks called chylomicrons. Chylomicrons are teeny tiny little particles consisting of triglyceride surrounded by a layer of phospholipids and some cholesterol and a hodgepodge of protein. So in the picture at the right, you see what amounts to being a tiny little particle. And that particle will essentially move through the lymphatic system and will ultimately get into the bloodstream. There is a connection between our lymphatic system and our bloodstream in our upper thoracic cavity, where the lymphatic vessels attach to our subclavian veins. And so these chylomicrons eventually do get into our bloodstream and will ultimately be transported to our liver or to cells of the body. And like I said, it's like a little delivery truck that this chylomicron will bounce around from cell to cell to cell, unloading its cargo of triglyceride. The only other component of the diet that we really have to discuss at this point is what happens to proteins. You'll remember that proteins are digested through almost a 2-step process. Step number 1 involves breaking down the proteins into polypeptides, which is accomplished through the action of pepsin, trypsin, and chymotrypsin, And then we're going to take those polypeptides and break them into individual amino acids through the action of carboxypeptidases. Once those amino acids are liberated into the digestive stream, they are going to be absorbed directly into the bloodstream and they will be transported through the body. And we can use those amino acids for constructing new proteins of our own. We can also use those amino acids as a source of carbon and energy for blood sugar regulation. You'll remember that not too long ago we mentioned a process called gluconeogenesis, the creation of new glucose, a problem that arises when our blood sugar levels start getting low and our liver basically bottoms out in terms of the amount of glycogen that's available, at which point the liver has to get creative and we use amino acids to create new glucose. The basic idea is if I take an amino acid and I remove the amino group, what I am left with is a 2-carbon fragment. 3 deaminated amino acids gets me a total of 6 carbons that can be the basis for me to make one brand new molecule glucose. In which case, during these periods of time where I'm fasting, either intentionally dieting or sleeping, my liver has the ability to provide glucose that my cells will need and require, even though my liver is running low on glycogen.